Hi, everyone. I want to welcome you all back to our 5th annual Big Give. As a reminder, our goal is to raise $50,000 in 24 hours to support our Ladder Learning Network. The Ladder Learning Network connects patients to care, physicians to physicians. We support clinical trials, and we also have our Ladder database, which supports information on our 215Q individuals and goal to create a natural history picture. So today I have with me Carrie Howell, our executive director, Dr. Judson, and Dr. Philpot.
Thank you, Naya. Thank you. Thank you both for being here so much. And you guys are some of our favorites that people ask over and over. We have questions for them. We want to ask them questions about UBE3A. So we're so excited to jump in, and thank you for just giving us a few minutes in the midst of all the work you all are doing. I know that you've done a lot of work on mouse models, and let's just kind of start at the top and go backwards just a little bit, at the top and backwards. Talk a little bit about, just give us an overview for people who are newly diagnosed families. Talk a little bit about the 15Q region and explain what this may mean to families who are newly diagnosed.
Sure, if you don't mind, Carrie, I have a few slides that I could share that help us get the discussion started. Here we go. Just a second. All right, can you see that okay?
Yes.
All right, and let me just go to this view. Okay, so what you're seeing here is at the top of the slide is a schematic of chromosome 15 as it might occur in a neuron. And what you may be aware of is that for most genes in your body, you get a copy from your father, so it's your paternal chromosome or paternal alleles, or from your mother, so that would be, of course, maternal. And as is the case for the 15q region, there's some really interesting parent of origin gene expression stuff that's going on here. So most of the genes in the region, like I just explained, you'll have what's called biallelic inheritance and expression. So most of these genes, like you'll see here, these guys in gray, you have expression of the gene from both maternal and the paternal copy. But there's something really interesting for a gene called UBE3A in this region, whereby in neurons, you're only getting expression of UBE3A from the maternal allele. So what that means is that in cases of duplication 15q, especially when it's inherited from the mom, you're going to have, in particular, overdosage of UBE3A amongst all these other genes in the region. And so what we think is, from our perspective, having some expertise in studying UBE3A, is that this kind of shines a spotlight on UBE3A as having some kind of particular importance to the way that DU15Q syndrome kind of unfolds developmentally and expresses pathophysiologically. So we wanted to understand through making mouse models how UBE3A overexpression by itself might be able to affect neurodevelopment and neurodevelopmental outcomes. And what you can see down here in this little cartoon neuron is a nucleus. And this is sort of just some data from another lab, from Art Baudet's lab at Baylor College of Medicine, where you can kind of see how this region sort of looks inside of a nucleus with a little bit of experimental magic. So these are some markers in red here over on the left that can show you inside of this kind of perforated outline of a nucleus where the maternal and paternal 15q region is. And what this green signal is this, it's an RNA, a long non-coding RNA from the antisense, which is actually silencing expression of the paternal copy of UBE3A. So really, if you just can imagine over here, this red region where there's no antisense, this is the region where you can get UBE3A expression from. And so if you duplicate this red region of the chromosome, you're going to get 2 copies of UBE3A along with, or 2 or 3, as the case may be for interstitial dupe or isodicentric dupe. You're going to get multiple copies of UBE3A along with multiple copies of these other genes. Whereas you were duplicating the paternal allele, where there's this antisense present, that kind of blocks UB3A. So you'd only be getting overexpression of the genes besides UB3A. So we think this has something to do with the difference in kind of phenotypic severity in paternal dupe versus maternal dupe 15q. And hence, as I mentioned before, why we're really interested in studying in our mouse model UB3A, at least for starters. So moving to this slide, this is something that I kind of alluded to before, where there's kind of differences in symptom severity as it's understood in the clinical, with the clinical picture of dupe 15q. So these are just cartoons of a sort of neurotypical karyotype of the region. So this is your 15q region of the chromosome, kind of in this colored area here. And you can see normal representation of that in this neurotypical karyotype. So this would be from a typically developing individual. And then here's cases where you would have interstitial duplication here in the middle. So you can kind of see kind of side-by-side duplication of this region within the same chromosomal arm of this maternal 15q. And also another scenario where you have what's called an isodicentric triplication. You kind of think of this as almost a kind of satellite chromosome where there's two extra copies of the 15q region. And so whereas this is going to duplicate your dose of 15q genes, including UB3A, this would actually constitute a triplication. And so generally speaking, in the clinic, what you're going to see is an increase in the symptom severity in the isodicentric scenario versus the interstitial duplication scenario. So this might include things like phenotypic domains like that have more to do with motor function, like hypotonia you see in younger children, also maybe delay in reaching motor milestones. And it also especially could affect some areas like intellectual disability and maybe most notably epilepsy and what's called SUDEP, or sudden unexpected death in epilepsy, which we're very interested in the lab and we know is of great concern for families due to the very extreme consequences of death during seizures. Very, very scary and very serious. With this in mind, we wanted to think of how we could best make a mouse model where we're overexpressing UB3A. So ideally, at the outset, we wanted to be able to recapitulate UB3A overexpression with interstitial duplication and isodicentric triplication scenarios in mind. And so what we did, I won't go into the details of how we engineered the mice, but we were able to do this, to make a long story short. And this is some data that really speaks to our success in this area. So what you're seeing here in this kind of top box is protein signal from the brains of our model mice and some of their neurotypically developing litter mates. So this is which we call wild type mice. So this wild type mouse has, in this given experiment, an amount of UB3A expression that kind of looks like this on our Western blot. And here are litter mates that are expressing one extra copy, which would be very similar to interstitial dupe 15q, and two extra copies here, which would be more similar to the isodicentric scenario that I mentioned before. So you can see this kind of stepwise increase in UB3A overexpression. And here is something we can do in the mouse pretty readily, which is actually get to 4 extra copies. And this is kind of a useful tool for us to really push the system and see if we can't really force phenotypes or push a neural circuit toward having deficits with tons and tons of extra UB3A. So we thought that would be useful. And of course, here on the far right is a situation where we have an Angelman syndrome model mouse, which is lacking maternal UB3A expression. And this is sort of our negative control to say, hey, our experiment's working, we're detecting UB3A, this isn't some other protein. But what's really nice to look at is the quantification, kind of the numbers that we put to experiments like this, where if we kind of set our wild type UB3A expression here at 100%, you can see that we really are accumulating in our +1 and +2, akin to our interstitial dupe and isodicentric cases, well in excess of 100% of UBE3A expression. So we really are creating an overexpression scenario that is, we think, relevant to the clinical picture of Dup15q syndrome. And so what can we do with these mice? Well, so far, we've used them to look at behavioral domains that are relevant, we think, to the clinical picture of Dup15q syndrome. So we won't go into details about exactly what these behavioral assays are, but we have different kind of tests and arenas in which we can test our mice to look for things like motor function. So, and I can give you just a brief overview of what some of these are. So we can kind of put mice in an open field and just track their movement over an hour to see if they're moving more or less than their wild type litter mates. We can put them on an accelerating rod to see how well they can maintain their balance as the rod spins faster and faster. This is a great measure of balance and motor coordination and their ability to learn to get better at that task. We can look at their cognitive function in different ways, how well they remember where they experienced a really scary stimulus, such as a little bit of a foot shock, or they don't really like swimming, as you might guess. So mice learn to escape a water maze. So they learn how to use cues that are on the walls to get out of that. And also we can look at different measures of their extinction of operant behavior, which is definitely more than I want to get into today. Of course, due to the manifestation of autism spectrum disorder in Dup15q syndrome, we wanted to look at how mice socialize. So this isn't an exhaustive test, but mice typically, if you give them a choice of interacting with just a regular object that's new to them or a mouse that's new to them, they typically want to socialize and be with a new mouse. And so we've looked to see how our UBE3A overexpression mice interact socially with each other. And also we've looked at how readily our mice exhibit seizures, especially when challenged with a drug that will push their neural circuitry toward a seizure outcome. And so what you can see here on the far right is just a basic summary of what we've observed in these mice. And somewhat surprisingly, we have yet to be able to push a phenotypic outcome in either the motor, the cognitive, or social domains, despite a lot of testing. Now, this doesn't mean that there aren't some subtle deficits in these models, in these domains, that might be teased out with further testing. But these are pretty exhaustive tests that we've put them through so far, and so far we've seen very little of a phenotype in these areas. Now, square that with the seizure domain, where we actually have seen some interesting things. So you can see this red check mark here, where we are seeing some impairments in this area. And let me just explain what that is a little bit. So when we test our mice, we're giving them a drug that sort of makes them susceptible to having a seizure. And we essentially measure how long it takes for them, the overexpressor mice and their wild type litter mates, to reach that seizure threshold. So how long it takes for them to start seizing. And what's very interesting is, despite extensive testing, we've never seen the UBE3A overexpressor mice seize faster or have more severe seizures. But quite interestingly, when we do evoke seizures in them and they have a kind of more severe seizure, what we call a tonic-clonic seizure, where they're losing posture, there's a risk for mortality when that happens, a risk for death. And they are much, much more likely, for reasons we don't yet understand, than their wild type counterparts to die in that very extreme situation. So we think that this might have some relevance to SUDEP, the sudden unexpected death during epilepsy phenotype that plagues especially some of the IDIC-15 individuals in Dup15q. So we're definitely following up pretty earnestly with that. But with this mouse phenotyping that we've completed so far in mind, and we're about to publish this very, very shortly, so you can be on the lookout for those data. We did, think about, okay, well, it's not squaring too well with the Dup15q clinical picture when you just overexpress UB3A. Is there any, sort of more relevant clinical population that we could compare our findings to? And it turns out, even though these individuals are quite rare, there is a family that's been described in the literature that seems to only have a UB3A gene duplication. So none of the other 15q genes, just UB3A. And it turns out in this family tree, when they inherit this mutation from either the matriarch in the tree or other females, that you tend to have some neuropsychiatric phenotypes, and they seem to be far less severe than what you see in Dup15q syndrome. But it does suggest that overexpression of UB3A itself could be doing something. But relative to Dup15q, it does seem like the other genes in the region are sort of needed to cooperate to produce the more severe Dup15q syndrome pathophysiology. So that's kind of where we're at in our research, is thinking about what we've just found with UB3 overexpression by itself. And we're starting to think about how other genes in the region might be kind of synergizing and mixing with the UB3A when they're overexpressed to cause more severe phenotypes. And so those are going to be some really exciting next steps for us in the coming months here in the lab.
Wow, that is so fascinating. And you did such a great job explaining that. For parents who are watching, what does that mean? Because I think one of the questions I get a lot is, well, when I see research like this, does this mean in 6 months something's going to happen? What does happen? When you're talking about the next steps in research for you all, what can the parents practically expect to happen with this type of research?
Yeah, I think there are a lot of different ways that this can go. So one of the things that's important to note is, although we're finding that UB3A by itself isn't a driver for a lot of these negative phenotypes, it doesn't mean that UB3A isn't important for many of the phenotypes. It just means that UB3A is likely important in combination with overexpression of other genes. Now, this can mean that in some ways, it can even broaden the therapeutic opportunity for Dup15q syndrome. So Dup15q syndrome could be treated by either downregulating and normalizing UB3A levels, or it might also be treated by regulating levels of some of the other genes that are in this region. So this is a very important area of research for us to understand. So what Matt and I and other labs like to understand are what are the other major players in this genetic locus on chromosome 15, which ones are really relevant to the disease pathophysiology. And with that knowledge in hand, then we can start designing therapeutic approaches. So an obvious one is just to downregulate UB3A, but are there other things that we can target, other genes and molecular targets that we can use for therapeutic opportunities? So it broadens our therapeutic targets and what we can actually do.
Oh, wow. Nika, I'm sure as a mom, yeah, go ahead. I saw you unmute. Go ahead.
I'm ready. Do you have any idea of what the other genes you might be targeting with UB3A?
Yeah, there are kind of some obvious candidates in the area. So, for example, there are a cluster of GABA receptors that are thought to be very important for seizures, but there's also this very intriguing gene called HERK2, which in many ways kind of synergizes with UB3A and kind of augments its role. And we think that could be a very important target.
Yeah, and just sort of from a practical perspective, one of the things we've looked at is just sort of what is the minimum size of the duplication, just, that's associated with Dup15q syndrome to kind of, help narrow in at least a little bit on this very long list of genes. And it just so happens that both the GABA gene cluster that Ben mentioned and also HERK2 are represented within that kind of minimal region. So we're intrigued by that for certain.
Yeah. Go ahead.
Were you noticing that the mice that had like 4 copies or, the max amount that you could do, that they were seizing faster or dying faster than those that only had one copy?
That is a fantastic question. And the short answer is we don't know that yet. We're actually setting up breeding to test that this summer. So yeah, we'll have to keep you posted on that, but it is definitely something we've thought about. We do know for some of the other domains that, such as sociability, that 4 copies doesn't seem to be any more deleterious than 2. Regardless of whether it's 2 or 1 or 4, we don't seem to get a sociability phenotype. But for seizures, we don't know.
But you do know the pseudo phenotype with 1 versus 2 copies of UBE3A, Matt, which might be worse.
Our data suggests that it might be a little bit worse with 2 copies, although statistically we can't say that yet.
I think that comes up a lot with our families, because we've seen families with 5 copies or some interstitials that have 2 or 3 interstitially. And so a lot of the times they ask, is the more that they have, is the symptoms more severe? And I don't think we've been able to answer that on any level in terms of symptoms. I think it really just depends on all the other genes involved. It makes me think, as a parent, I'd be really curious if we were able to get, our goal is to get 1,000 of our families into our ladder database, showing their symptoms, their severity of symptoms, and their genetic variant of 215Q. Would that help if we had a big data set of all these families that have the different extra copies of 215Q and all their symptoms? Would that help in your research as well to see what the next step might be in the mouse models?
I think that's extremely helpful. So if we start to see what we call a genotype-phenotype relationship, that tells us domains where we should focus our lens. So for example, if you see enhanced seizure phenotype with 2 versus 1 copy, or enhanced motor phenotype with 2 versus 1 copy, then that encourages us to look in those domains to see if there's something analogous in the mouse models. So it really is important, and that's the type of information that guides our research.
Yeah, and also with,
Go ahead.
Sorry about that. Yeah, so I think there's some really exciting work coming out. Basically, it seems like monthly at this point, from folks like Eric Levine and Stormy Chamberlain, looking at the patient-derived neuron models, and while they might not be able to phenotype their system, these cultured neurons to the same extent that we're able to obviously phenotype mice behaviorally. I think they can develop a very rich sort of collection of cells from different patients, and that might actually help provide some more insights as well, especially if there's really good registry of the phenotype genotype relationships.
I'm curious and I love the word you said a while ago, Matt, just practically speaking. So if a family hears this and they're like, oh, this sounds exciting, going back to the idea of what does practically happen next? Does a pharmaceutical company see your research and say, we're interested in talking with families? Like, go through just a 2-minute on what happens to this research when you find solutions and the right answers, because a lot of our parents really do think, oh, this is hopeful, and in 6 months we'll have a therapeutic treatment available. Can you talk a little bit about that?
Ben, did you want to?
Sure. I'll give it a stab. I mean, I think what's very difficult to predict is timeline, how rapidly things can occur. And I know that's frustrating from a parent perspective, because you're ready for a treatment, yesterday. But from a scientific perspective, it can really, the timeline can really vary. So with the type of information we're getting, we're now kind of focusing our lens on different therapeutic opportunities, but then we have to try those out. And the way we first try those out is using animal models, because we really want to consider safety. So before anyone's child is given a therapeutic, we need to vet it as thoroughly as we possibly can in cell-based models. So for example, the patient-derived cell lines, and as well as the animal models of 215Q syndrome. So what we'll do next is, both within our own lab, we're trying to develop therapeutic opportunities, but we'll also work with pharmaceutical companies if they reach out to us, and I'm sure they're working obviously independently of us as well. But we're all trying to work off the same type of information. So anytime something is published in the public domain, it goes out to everyone, and it kind of gives them the same starting points of what to look at and where to focus their lens. So now we, of course, know that UBE3A is a target, but this broadened our lens to look at other targets such as HERC2 or some of these GABA receptors, and try to figure out ways to normalize the expression of those genes or their downstream targets, and to help treat Dup15q syndrome.
That's so helpful. It's so helpful because I think families do get frustrated and they go, why isn't the Alliance speeding this up? And we're like, oh, we wish we could do that. I want to go back and do a 30-second, probably less than 30-second commercial. If someone's watching this and they are like, what are you talking about, enroll in Ladder? Reach out to one of our team members. We will help you get enrolled. I love that you all said how important that is, because we know when someone's recently diagnosed, they'll call us and go, you need a lot from me. Like, you want the genetic reports, you want this. And we're like, I know we just met you, give me everything you have. But we want them to understand that this is the future of therapeutics, is to have that information in Ladder. So I love that you all talked about that. And I do invite anyone who watches this to reach out to Neka or myself, and we'll get you connected with how to enroll, because it is really the key on our end of what we can do now. So thank you. Neka, what other questions?
Well, can I actually expand a little bit on the
Please.
importance of doing Ladder, because there are multiple ways that helps. First of all, it's kind of a free way that families can help. So if they don't have means to donate money to the foundation, this is something they can do for free and get involved in that. And it makes a huge contribution. So as scientists, we can take the genotype, phenotype information that you get and figure out how to guide our experiments. So it really helps us. But it also encourages pharma companies to get involved in trying to develop therapeutics for Dup15q syndrome. And it helps for many reasons. One, if they realize that the population is big enough, they realize that it can make a return of investment. So pharmaceutical companies don't want to invest in something that's so rare that if they put in millions and millions and millions of dollars, that they want to at least be able to, at least kind of break even. So it encourages them in that sense. It also makes a clinical trial much more feasible, because it means that you have a patient registry, you have all kinds of information. So when therapeutics come along, it means you can reach out to the patient population, get them enrolled in the clinical trials. You have a good idea of what outcome measures to be, where you might be able to move the needle with a therapeutic. And this is really useful to the biotech and pharma companies, and helps get them involved. And they're pouring their money into efforts in addition to kind of the academic money that's going into an effort, just increases the chance of getting a therapy. So it's really important to get involved and do things like contribute to Ladder.
We didn't even pay you guys to say that, but that was so well said. That's exactly what Neka and I have been preaching, is how important that is. So, wow, well said, well said. Thank you. Neka, do you have other questions or comments?
No, I'm just really thankful for all the work that you guys are doing and helping make a better tomorrow for our kids. I know it's a long road, but we appreciate all the hard work that you do. And I always look forward to talking to you guys every year on our Big Give.
It's our pleasure. It really is a lot of fun.
Yeah.
The foundation's been great.
Thank you. And thank you, Dr. Judson, for explaining it so well about the UBE3A, because I can't imagine often when I look at our new families, and I think of one that has a baby that was diagnosed, and just, it's so overwhelming. And then you're thinking, what the heck, what is, I just found out, and now I'm learning all this. So I love that we can offer these resources to them as well. So thank you both. And thank you for what you're doing.
Glad to help.
Pleasure.
Thank you. All right, Neka, you want to close this out?